Okay. All right. So the very first thing that everyone should do is look at conversions. So remember, we use SI units. I'll probably ask you in the midterm what an SI unit is, or maybe ask for examples. So remember that the SI units are based on the metric system, and they're based on the number 10. So I would definitely practice doing this decimal movement stuff. So let me give you guys an example of this real quick so that you know how it's done. So I'm just drawing here. Okay, so basic unit. So basic units are things like meters, grams, liters, and then you add these prefixes in front of them. So say we have 3 meters, 3 meters, and we're trying to convert it to centimeters. So there are 100 centimeters in a meter. So we could just multiply 3 by 100 and get 300, or we could take 3 meters, so 3 meters, and move the decimal point over twice. I mean, yeah, twice for centimeter, because centi is 100, so that would be 10 to the second. But if you're gonna convert 3 meters to millimeters, you would move it over 3 times, and you get 3,000, because milli means thousands. Or again, you could just multiply 3 by 1,000. And as you've seen, the number gets bigger if you're going in this direction, and it gets smaller if you're going this direction. So if I was to ask you, convert 1,000 meters to kilometers, you would move over once, twice, 3 times, and you get 1 kilometer. So the number got smaller when you go to a larger unit. The number gets bigger when you go to a smaller unit. Make sense so far? Okay, I also might ask you to convert between, oh, hold on. Does it say it's recording? Because I need to upload this for everyone else. Let's see. Let me stop sharing for a second. Yeah, it is recording. Whew. Okay. Anyway, back to prep. Okay. So I might ask you to convert between US customary units to metric, or between metric units. So let's take, for instance, 12 inches to feet. So there are 12 inches for every foot. So the way we would convert 24 inches into feet would be by multiplying. You always put whatever you're trying to get rid of. We're trying to get rid of inches at the bottom. So inches go on the bottom. So there are 12 inches for every foot. So that's a 1. Sorry, it's kind of hard to use this pad. So you will multiply 24 by 1 and divide by 12. You get 2 feet. Inches crosses out. Yeah, so every time that you're trying to get rid of a unit, you put it in the denominator, and that's how you get rid of it. Okay. So you will be asked to do conversions like that on the midterm, and yeah. Another thing that you guys have had issues with are the labeling graphs. Okay, so remember, you have a caption. You can have a caption and then a title. Sometimes I call this the title because you really don't need a title up here, but usually the caption goes at the bottom. Make sure that I'm going to have you guys create a graph. I'm going to make you submit it through Dropbox. I'll send you that and you'll have a few minutes to turn that in. So once you're done, you're going to draw a graph for me and you're going to submit it to Dropbox. So make sure that your graph caption or title is at the bottom. Then you have some sort of scale. You have a axis label, unit. Then you have your different treatments. And so you have those labels and a subheading. So this explains what these are. Okay. All right, so I need you guys to remember a couple of things. So line graphs, the only time where you would use it would be for continuous, that's too fat, continuous data. And bar graphs are for discrete variables. Okay, so continuous data, it means that, let's say these are days, and this is let's see, let's just call it temperature. Don't mind the scale for right now. But what if you want to know the temperature on the 15th day? You can just find a point there and essentially predict what would happen. So there's an infinite number of points that you can reference in a continuous graph. That's why we have a line connecting all these points, because it's continuous. If you were to swap day 100 for day 30, if you just swap them, it would make no sense because you're moving this data that's dependent on this data. So that's why we call it continuous, because the points, the individual points are related to each other in some way. Discrete data usually describes things that are in groups. So you have different treatments. Those are the different groups. There's discrete variables, and that's when you use a bar graph. Okay, so another thing, remember, the thing that you're measuring is a discrete, the dependent variable. Dependent. The different treatments are the independent. So dependent variable, independent variable. The dependent variable goes on the y-axis. That's the vertical axis. And the independent variable goes on the x-axis, or the horizontal line. Okay. So far, any questions? Can everyone hear me, by the way?
Yeah, we can.
Okay. All right. If you see this symbol, going back to conversions, this is a mu. That just means a millionth of a whatever. So that's a millionth of a liter. So it's a really small unit. That's, I think it's 10 to the sixth. So to the minus 6 actually. So it's a millionth. So if you were to convert from 3 millimeters, you would go to the right 3 times. 1, 2, 3. So you would have 3,000. But if you had, let me make more room here. If you had, let me just erase all this. If you had, if you had 3 meters, it's terrible, meters, you would go 1, 2, 3, 4, 5, 6. So it would be 3 million micrometers. Again, it all depends on where you start. So if you're to convert this micro whatever is way out here, so it's right, 1, 2, 4, 5, 6. Yeah, so it's way out here. So if I ask you to convert from micrometers to meters, you have to remember that you got to go through these steps. So you're going to go 6 to the, well, if you're starting from micrometers, you got to go 6 this way. If you're starting from meters, you got to go 6 that way. But maybe I'll do milli to micro, and then in that case, you would only go over 3 times. It all depends on where you're sitting. I would remember that, what's it called? King Henry died by drinking chocolate milk, and then micro would be also on there. So usually we don't use deci. It's not a useful unit. We don't really use deca or hecto. They're not that useful. So the most useful ones are kilo, centi, milli, and micro. And remember your basic unit: meters, grams, and liters. Okay. So that would be part of chapter 1. Okay. So, okay, let's see. The next part, if you guys are using your lab manual on chapter 1, page 26, there's a description of accuracy and precision. So remember, accuracy is how close you get to the actual or true value. So if we know that this pen is 5 inches and your measuring tape says it's 4 or 8, it's not accurate. If your measuring or your caliper tells you that it's 4.5 and you know it's not 4.5, and you measure it again and it gives you 4.5 again, and it keeps giving you the same thing, that means it's precise because the measurements are close to each other, but not accurate. Of course, there's levels of accuracy and precision. So if you have like, wrong thing, you have 6.5 being your true value, and you have things like 6.3, 6.4, 6.7. So you're somewhat accurate to a couple of tenths places. But if you're like say 7 or 8, then you're way off, and you're losing accuracy the further away you get from that true value. Precision, you can have precision. You can have a bunch of 6.5s multiple times, or you can have a bunch of 6.4s. So the closer these things are to each other, the more precise you are. Okay. But you can also be completely not accurate, but still precise. I would review those terms on page 26. Okay, let's see. Oh, volume readings. Okay, well, let's, on page 32. Okay, so I told you guys about tearing. Tear or tearing. So remember I told you that a lot of things you have to use like some sort of vessel to put your sample in, and you have your scale, but you don't want the mass of whatever the container is. So you put the empty container in there first, click the tear button, and then that removes it. And so you're only weighing your sample. You can also do this on an electric scale by first weighing out the cup or whatever it is, writing down the mass of it, and then when you get your final mass, you subtract it. It's exactly the same thing. So tearing would be a term. Page 33, where it talks about volume readings. So you have graduated cylinders. Usually they're always in this form. They're always in ascending order as you go up. So this is a graduated cylinder, and whenever you pour liquids in it, usually things like water have like a high surface tension, and they will create this thing called a meniscus. So MENISCUS. Okay. So I told you you have to read at the lowest point. So that would be this point here. So you get an eye level here and you read this point. So some people were reading up here. That's not correct. So on page 34, you'll notice that pipettes, a lot of times they have 0 here, and then 1, 2, 3, 4, 5. If they're a 5 mil pipette or a 10 mil, so they're backwards, right? Because a lot of times it's more useful to know how much fluid you've already pipette into your sample than how much you have in the pipette. So I told you guys last week to pipette all the way up to 0 and then pipette down to 3, I believe. So you pipetted 3 mils of water, or I'm forgetting which class it is, but anyway, you pipetted 3 mils of water. And we really don't care about what's remaining in our pipette. That's why usually it's in this direction. So I might have to ask you about telling me how much is left in here for one of the questions. It's like a higher order. It requires you to think in a higher order. Okay, so calculating temperature. Okay. So The formula for converting from Celsius to Fahrenheit is, let's see, we have 0 Celsius here, and we're going to convert to Fahrenheit. So we're going to multiply by 9/5. Then we're going to subtract, we're going to add 32. So this, of course, is 0. Add 32, you get 32 degrees Fahrenheit. The way I remember which numbers to use here is we take Fahrenheit and we do the exact opposite. So let's see, we do the exact opposite. So we take 32, we subtract 32 from it. Maybe I should use a different number. So let's say we have 50 degrees Fahrenheit. We subtract 32 from it. What is that, like 18? Yep, we get 18. And then we multiply that by the reciprocal of this, 5/9. And whatever that is, 18 times 5/9 equals 10 degrees Celsius. Looks right. Thumbs down. So yeah, if I'm going towards Celsius, which is a smaller number, the smaller number goes on top, the larger number goes on bottom. If I'm going from Celsius to Fahrenheit, which is the bigger number, I put the bigger number, which is 9, on top. So that's how I remember it. Okay, so there are post-lab questions there. I would try those as practice. Okay. All right. Appropriate tools to use for these things. Shoot, come on. Let's see. Okay, so what would you use for mass? You would use a scale for maybe the width of, let's see, your finger. We would use calipers. For the volume of a cup, we would use something like a beaker. If we were to measure the amount of I don't know, what's the small thing? Well, the only times you would use a beaker is when you're measuring a large volume, because they're usually larger. If you're using a smaller, if you're measuring a smaller amount of fluids, you will use a graduated cylinder. That's the skinnier one. So graduated cylinder, beaker for larger things. Okay, so moving on to the second chapter. So are there any questions for the first chapter? I guess that's a no. Okay, the scientific method. Okay, so what I would do is go through the steps of the scientific method and highlight the most important part of that. So observation. Okay, let's see. Observing a phenomenon, an aspect of an organism, or discovery of a problem. So anything that you observe, anything that you see, question, So yeah, I would just highlight the most important part of each step in the scientific method on page 49 through uh, page 55. So you have 7 steps. Okay, yeah, also parts of the experiment. I got to go back to my graph. Okay. Well, maybe I'll just do a different graph here. Okay, so let's say we're measuring the length of a plant, but we just bought some fertilizer called fertilizer X, and we want to know if it's more efficient than fertilizer A or just regular ground or regular soil. Okay. So in an experiment, you have experimental groups. So these are the experimental groups. And then you have some sort of baseline. So this is like the original, no treatment. This is the one that doesn't get any different treatment. Because what if we use these two experimental treatments and they're really not different than just regular soil? So we also have to compare these experimental groups to the control group. Move this here. Okay. Yeah, that's terrible. So yeah, this is the control group, just soil. So we have soil. Um, okay, so we have soil, we have fertilizer X, and then we have fertilizer A. Okay, so if we were just comparing these two, we wouldn't know that fertilizer X is bad for the plants. So this would be, okay, so remember, these are the treatments, so this is the independent variable, the dependent variables on the y-axis. So this would be like plant height in inches or whatever. So yeah, experimental groups and control groups. So remember, there's also something called a control variable. And this has nothing to do with the control group. So something that we can use as a control variable would be the same amount of water. So what if we gave group X less water? Because obviously that's going to change the outcome. So we use the same amount of water, same amount of sunlight. So we leave the pots out the same amount of time. Anything that could maybe change the outcome or the results that we don't want to change or we want to keep the same are the control variables. Okay. So I'll probably have you guys identify control group, experimental group, control variable for the midterm. Okay. Okay, I guess I got to talk about hypothesis. Let's see. Okay. All right, so if you look on page 50, so a hypothesis is a potential explanation to the question. So a lot of you are telling me, you're giving me the prediction, really, rather than the hypothesis. Remember, the hypothesis is a statement. So please don't put I think or I feel. Tell me this is going to happen because of this. So let's see, let's think of an observation. So we see that sunflowers turn towards the sun during the day. So they're, or wait, let's scrap that. Let's say that sunflowers turn to move around during the day, and we don't know why. I mean, we all know why, but what if we didn't know that, and we're trying to figure out why? Okay, so that's our observation. Sunflowers move around during the day, face different directions during the day. So that's our observation. Our question would be, why do the flowers move around during the day, but not during the night? And we can think about the reasons why. And so we look through the literature, and then we find out something about the sun feeding the plants. Like, I know we all know this, but it's just for the sake of this question. So now that we know all these things, we can form a hypothesis. So we're going to say sunflowers face different directions because they're following the sun as it moves through the sky. And so we have that hypothesis, but now we have to prove it. So we have to create a prediction. So we can't just say that and have no way of testing that hypothesis. So we tested by saying two things. So remember, it's an if-then statement. So you say if the sun is out, then if the sun is out and emerges in the east, rises in the east, and sets in the west, the sunflower will face east, then face west throughout the day. So that's our prediction. So the prediction is just what you think is going to happen during the experiment. So yeah, I would use that if-then statement format. So again, the prediction is assuming the hypothesis is correct. Because sometimes you guys give me a hypothesis and the prediction contradicts the hypothesis. So yeah, no. Okay, so moving on. Okay, we're still talking about experiments. So I told you guys we need to do replications in science a lot of times because we need to find out the truth. And the reason we do that is because, remember, we have large populations and people are different, animals are different. So you can have all sorts of variation. It's terrible. Okay. So you can have this group, you can have this group, this group, this other group. But if you're conducting an experiment and this is your sample size, it's not very representative of what's going on in here. So you need larger sample size so that your conclusions about your experiment are more accurate. Because what if we said, let's buy a bunch of t-shirts for Georgia Southern students, but we don't know how many small, medium, or large we're going to need. So let's just go out and get a sample of students. But I'm lazy, so I'm just going to go to Hanover Field House and just ask the people there how large, what their size is, and we just ask basketball players. They're all going to get larges, and so the school is just going to buy a bunch of larges, and then everyone is going to be like, "What the heck? I'm a medium or I'm a small." So you need to get a large sample size to be able to make a decision or something like this. So we need large sample sizes in order to do um, more accurate studies. Okay. Okay, scientific writing, um, page 61 through 63, I think. Okay, just look at what the most important part of those definitions are. So what can we find in these things? So um, let's see, what is the abstract? It's the one paragraph summary or the most important parts of the entire paper. Introduction, you have the background information that you need to understand the study. Um, the importance of the study, so they're trying to sell you on the experiment that they're doing. Um, it might tell you the research question, what they're trying to find out, their hypothesis, what their objective is, the materials and methods. That's just a detailed, essentially like a recipe of what they did in the experiment, the results. Usually that's graphs and tables, and they tell you what happened in the experiment. Discussion, that's where they talk about what the results mean. Um, conclusion, that's just the big picture stuff. Let's move on to chapter 3. Okay, so chapter 3 dealt with bacteria, right? Okay. All right, so bacteria, they're single-celled prokaryotic organisms. Okay, some of these are pathogens, but not all of them. So pathogens, that just means that they are disease-causing agents. So some bacteria are, some fungus are, some viruses, well, a good bit of viruses are pathogens. So anything that causes disease is called a pathogen. Okay. But some bacteria are good for bioremediation. So bioremediation. So bio just means living. Remediation is to fix like a situation. So we use bacteria to treat sewage. So they break down all the number twos and all that goes into the sewage. They also clean up toxic substances in water and that stuff. There's also something called a probiotic. So these things are found in food. So they help with digestion, they make you feel better. They also make uh, it easier for you to digest a lot of food. So we already have a lot of bacteria, and these things just help digestion. So bacteria are like single celled, right? So they're very tiny, they're microscopic. So usually we are not able to see that with the naked eye, so we see their colonies. So these are like aggregations of thousands of bacterial cells. Okay, so let me quickly show you guys a thing that I think I glossed over last week. Okay, can you guys see a PowerPoint? Okay.
I can.
All right, so okay. All right, so bacterial shapes. So we have these spherical ones, they're like little balls. Those are spherical. Um, the Latin name for it are coccus. Then we also have these rod-like ones. Those are bacillus. Then we have the spiral ones. These are spirillum. So those are the basic shapes of bacteria. Yeah, remember we were rushing through this, so I don't think many of you got the chance to see that. Okay. The different types of disinfectants, and let's see. Where is it? Where is it? So anyway, let's see. Let me share this other one. Okay, so antibiotics. So these chemicals kill bacteria and fungus in the body. Then we have disinfectants. So these are used for non-living surfaces. And then we have antiseptics. So this is for living tissues. So remember I told you antibiotics, those are things like ampicillin or penicillin, anything that you take in like pill form to treat some sort of bacterial disease in your body. Disinfectants, these are things like hand sanitizer, which is kind of an antiseptic, but a more um, accurate example for this would be like bleach that you use to like clean counter surface or a desk or your phone. So it would be like bleach wipes and bleach. Antiseptics, those are things like alcohol, like rubbing alcohol for like a cut, or like hydrogen peroxide. So this would be for like a cut on your skin. So again, these are for living tissues. Okay, how are we doing on time? Okay, we're doing okay. Okay, this diffusion method. So I told you guys, well, we did it in the lab. We used these small discs in the middle of the plates, and then we measured the zone of inhibition. So it's the diameter of this empty zone right here. That was the zone of inhibition. I may ask you guys about food safety stuff, but it'll be open-ended so that everyone can write whatever they want. I just want to see how you're thinking. Yeah. And yeah, it should be pretty easy to get those points. Let's see. So chapter 4, I think this is the last chapter. Yep. Last chapter that we covered. Okay, so back to the PowerPoint. Let's see, where are you? Okay. Okay. Okay, I told you guys about the different microscopes. So the light microscopes, those are the ones that we used in the lab. So those were the compound microscopes, dissector microscopes. So the parts, remember ocular, meaning eye. So this is what allows you to see in there. You have the objective lenses. They have the stage, light source, condenser. Then we have stage controls. These are the things that move the stage forwards and backwards and side to side. Then we have the coarse focus knob. This is what moves the stage up and down, changing the it's called working distance. And this is what allows us to focus the image. Then you also have the fine focus knob. This is the smaller of the two knobs. Usually we show the other side because this knob is sticking that way more further out on the left side than on the right. So yeah, I would look at those, the parts of it. Okay, so anyway, magnification, the way it works, we have the ocular magnification. Usually it's 10 for most of ours. And we have objective lenses that are 4, 10, and 40. So again, remember, we multiply ocular magnification by objective. So 10 times 4, you get 40, and so on. So remember how to do all that. So that's the compound microscope. And remember, this one inverts images. So it flips things around. So remember, we saw E's that were like this with a naked eye, and then when we looked at them under here, they were backwards. Yeah, they were backwards and bigger. And whenever we move them one way, then we're actually moving the other direction. So it inverts the image. This one, the dissecting microscope, you'll use it for like larger things. Like you can use it for looking at live like bugs or like a large tissue. While this one we would use for looking at small microscopic things like cells. And this one doesn't invert the image. Okay, scanning and transmission microscopes. Okay, another thing that I forgot. Remember, this one creates a 2D view, so it's a flat image. Uh, the dissecting one creates a 3D image. Then you have the SEM. So this one scans the outside structures of the image. So you get a 3D image, and the TEM makes a 2D image. You can look inside of cells, so you can look at different layers of the cell. So this flat, tiny microscopic thing is the compound. This larger 3D image is the dissecting. This is the scanning. And this one is the transmission. So this one just scans the outside and it's also 3D. So these are 3D and these are 2D. Okay. Uh, okay. Okay, so we have different types of slides. So prepare slide, these are the ones that were fixed in resin, so you can keep them for years and years. That's one of the benefits of a prepared slide. A wet mount, usually a benefit of the prepared slide is that they last a long time. A negative thing about prepared slides is that you have to kill the organism to fix it in there. The wet mount, you can look at living things. That's the positive about it. But the negative about it is that you can't store it. It's a wet mount. It's gonna dry out if you don't fix it. Okay, so the reason we use stain is to create contrast, to color specific structures or body parts of the specimen. Um, I don't think I'll ask any questions about that specifically, so I think we should be done.
Is this PowerPoint going to be on Folio?
No, they don't allow us to share that stuff, but I will upload this video since I'm recording it, so you can watch it again.
And are we allowed to use calculators?
Yes, yeah, I need to make an announcement about that. So let me show you what's allowed. So Okay. So a four function calculator. Can you guys see the calculator that I just pasted on my screen? So it has to be one of these. No scientific calculators. You really won't need to do that many calculations. Well, you will have to do a few calculations, but really all you need is this. You don't need a graphing calculator, no scientific calculator. Just, and also during the test, you can't be wearing any electronic devices. So please no iWatches, any smartwatch. Um, you can't be on an additional phone. You have to use your phone to record yourself. Like, I guess we'll have to do like a quick run right before the exam actually starts, because I want everyone to be able to take the exam. So yeah, so I can see your screen and yourself, and I can see your area so that you're not on notes. Yeah. Yeah, that's pretty much it. Any questions? Any questions? No? Yeah, I would look through those post lab questions at the end of every chapter just to make sure that I know what to expect. So yeah. If you guys don't have any questions, I will also have office hours like in the hours leading up to the exam. So I guess from 2 to 4. So we can like look at your setup for the exam. If you have to be in class right before you come to the lab, if you have a laptop, bring it and we'll just proctor your exam in class. So I don't really expect there to be any Wi-Fi issues if people come to the lab, but they just told us just in case there are Wi-Fi connectivity issues. We'd like people to do it from home or somewhere nearby so that they don't all have to be using the same Wi-Fi. But I think you can do it from class. That way you don't have to do all these other things and be scared that you're going to be locked out of the exam, because that will eat into your time. So if you guys want to come into the lab, you're welcome to do that, and I can set you up. Any other questions?
If we come to the lab, do we have to video ourselves still?
No.
Okay.
Yeah, if you come to the lab, I'll just be proctoring, so I'll just be looking at your screens. So yeah, I think, yeah, everyone else has to be videotaped, and I'll be recording your Zoom. So basically, this is how you're going to be taking the exam. Usually we had other software, but it didn't work well at all. So, yep. If there aren't any questions, I think that's it. That's pretty much it. No? Okay. So I guess I'll see you guys on Monday. Bye.